whole genome sequencing at birth, consenting adults, sequencing babies. The hashtag for this event is Genomics Conversation. And this event has been made possible thanks to the kind support of Genomics England. Genomics England's Newborn Genomes Programme will co-design and run an ethics-approved research pilot embedded in the NHS to explore the benefits, challenges, and practicalities of offering whole genome sequencing to all newborns to accelerate diagnosis and access to treatment for rare genetic conditions. And in the vision document created by Genomics England for this project, it says the programme will aim to evaluate the utility, feasibility and impact on the NHS of screening for a larger number of childhood onset rare genetic conditions in newborns, including, and I think this is an important bit, what support they'll need. Second, it will aim to understand how, with consent, genomic health data could be used for research to enable new diagnostic discoveries and treatments to be developed. And also, it aims to explore the potential risks, benefits, and broader implications of storing an individual's genome over their lifetime. And this NHS research pilot will align clinical care with research and will feed back insights to parents through the NHS within a research protocol. And it's crucial that these research aims are carried out while maintaining public trust and while considering the impact on NHS clinical care and workforce. And it's probably never been a period in our history when we've been more concerned actually about impact on the NHS staff and the clinical care. And Genomics England say that they work to ensure that their work is human-centred, ethical and translatable into a clinical service. Now, PET is producing 3 more events after this one to help inform this programme, to allow plenty of opportunity to discuss the special sensitivities that a project of this magnitude may give rise to. So tonight's panel are going to be focusing on consent. So we have Amanda Pichini, Dr. Simon Ramsden, Waheeda Abbas, and Professor Anneke Lucassen. And so it gives me great pleasure now to introduce Amanda Pichini, who is a consultant genetic counsellor at Genomics England and a member of the Newborn Genomes Programme's NHS steering group. She has previously worked at the West of England Genomic Medicine Centre, delivering the 100,000 Genomes project, and she has worked with Health Education England's Genomics Education Programme.
So some of you may have attended the first session in this series held a month ago, where we talked about how to decide which conditions to include in the initial screening panel. And inevitably questions came up around consent there as well. So just to reiterate, as you've heard Sarah already describing, the Newborn Genomes Programme will involve a research pilot of whole genome sequencing in newborns, embedded in the NHS and alongside standard of care newborn screening, such as the blood spot test, with an opportunity for careful evaluation to understand how and whether this can be delivered. At the crux of this, and the focus of our discussion this evening, are parents making a decision about whether or not their child would be part of this programme. And I should also say that rather than hearing me say consent, my preference usually is to describe this as an informed decision or an informed choice, because I think really that better captures what's needing to take place. So I think there's 2 key aspects to this. A choice to receive information about a number of rare conditions, let's say 200 more or less that are actionable in early childhood, and a choice to contribute de-identified genome and health data to Genomics England's trusted research environment, built for the 100,000 Genomes project and now for patients in the NHS as well, and which can be accessed by approved researchers, both academic and industry, for health purposes. And with this, the opportunity to decline or to withdraw at any time without other care being affected. There's many operational and ethical issues to consider in this particular area, so I think we're really fortunate to have opportunities like this to understand and discuss how we can design a programme that enables parents to make an informed decision, and continue to learn throughout the pilot on how we can improve on this. So when we think about consent in other areas of medicine, there's often 3 elements required to ensure that it's legally valid. That the decision is made voluntarily and without coercion, that the individual making the decision has the capacity to consent, or in the case of newborns, that this is done by someone with parental responsibility, and that the individual has sufficient appropriate information to make a decision. So all makes sense, but that third one in particular can feel quite subjective. Genomics isn't entirely exceptional to other areas of medicine, but we do have to think about some unique considerations that this brings to the table around consent, including the many possible outcomes of a genomic test, the potential uncertainties and complexities around the meaning of genomic information, and implications for the present and future for the individual and their wider family. This can mean that very specific consent is difficult to achieve, but doesn't mean that it's not valid or that an informed decision couldn't be made. I think a lot depends on the context. Whole genome sequencing is just the technology. It's what we do with that, what we're feeding back, and what it will and won't tell us that's important to understanding what someone would need to know to make an informed choice. Part of this does mean then thinking carefully about what would be fed back, and there's reasonable questions to think through around this, particularly given the fact that we have the potential to find out about a person's future when they're a newborn and when they can't make this decision for themselves. So while we would have the whole genome sequence, we can be targeted about the results we're returning and limiting as much as possible the potential to uncover things that are unexpected, conditions where the onset or treatment is exclusively in adulthood, or where we would be really unclear about what that gene alteration would mean. What else is important to think about regarding the context of this program and being able to make an informed choice? So as a genetic counselor, I'm accustomed to speaking with patients and family members about genetics and genomics, whether in the context of their family history, adapting to a condition or risk, or supporting someone to make a decision about having a genomic test. Typically, for most genetic counselors, these discussions take place in a specialist service where someone's been referred because they're suspected to have a rare condition, or we know about an alteration in a gene in someone's family that we can test that person for to clarify their risk. So the chance of finding something is relatively high. And part of our interactions includes understanding where each person or family is coming from. What have their experiences been? What's their current understanding? What are their worries, their hopes, their expectations that are the driving factors behind them making a decision? In this program, we'll need to think carefully about how we can use elements of this approach to ensure an informed decision can be made in a public health context, where the volume is high and the chance of any one newborn having one of these rare conditions is very low. For most babies, as is the case with current newborn screening, we won't have a high suspicion of finding one of these conditions. This means it's different to situations where a parent is motivated to find answers for their child presenting with certain symptoms, and may therefore be more motivated to continue and contribute to research. Now, again, this isn't entirely something new. There are screening programs that happen in the antenatal and neonatal period that healthcare professionals such as midwives are experienced in guiding parents towards making a decision that's right for them. And I'm sure Waheeda will discuss her thoughts on this shortly. But we have to recognize that parents may be coming at this decision with different perspectives regarding the social contract that exists between the public and the health service. We do have some good sense of what genomics will change or add to the social contract that we have between public and health services. And this image is from a public dialogue in 2019, which highlights the importance of genomic research to provide benefit for others, as well as some red lines for how genomic data should be used. A final consideration on context is that genomic testing typically sits across clinical care and research. For example, when we find an alteration in someone with a rare condition, it can be difficult to know whether that alteration is actually harmful without relying on some level of research to understand this better. So research is, in essence, needed to ensure we're providing safer and more accurate clinical care. I think we have to accept this as an inevitability with genomics and be able to convey this to patients or parents in a way that helps them to appreciate that we're continually learning about genomics, and therefore our understanding and knowledge could change over time. However, while we're delivering a research pilot to understand whether whole genome sequencing is useful in this newborn context, we do need to make sure that we have a process set up to make clear that this is research at this stage and a separate decision to one that's being made about the current newborn heel prick test. This could mean, for example, thinking carefully about who's having the conversation with parents and how the information we provide about the program makes this clear. A question that often comes up in the context of consent in genomics is, how long is this discussion going to take? And particularly, if we're talking about 9 conditions in the current newborn screening test now, and this is often an extremely brief discussion, how on earth would midwives find the time to talk about 200 or more conditions? Now, we'll be exploring how to do this best over the course of the pilot, but I would argue that an informed decision is not made by taking someone through all of the conditions that we might look for. I would argue that what's most important to enable informed decision-making, to pick one word, is trust. Trust between the patient or the parent and their healthcare professionals in the antenatal and neonatal period, and trust in the organizations that are offering such a test, that their information is going to be kept and used safely. Easy to say, but not always easy to achieve, of course. So how do we achieve and practice trust? This isn't an exhaustive list, but a few things that I think are really important and will hopefully help with our discussion. First is being honest about uncertainty. Regardless of how targeted we are in our analysis, there's always going to be a level of uncertainty and nuance when it comes to genomic testing, as there is in other areas of medicine and health. I can think of many situations in clinic when a parent is asked what that genetic result is going to mean for their child's future. Will they get one of the complications that they've read about? And often the honest answer is, we don't know this at an individual level. Uncertainty can have a negative connotation or a positive one, and balancing feeling a lack of control with potential hope for the future is really important. And in that sense, we need to be honest about this, both when a decision is being made about entering this program, as well as when any results are being fed back. Secondly, to clearly communicate and manage expectations from the test, whether initially as a research pilot or in any form it may take in the future. And this means understanding what parents' expectations are in the context of everything else they have going on as they're having a baby. And we can communicate how long the results will take and what the test will and won't tell them about their newborn. And not to be flippant about exact amounts of time, but just as an example, I think by spending 5 minutes gaining an understanding of parents' expectations, worries, and hopes about this test, and by being curious and inviting, you can make more from the subsequent 5 minutes discussing the test and its key implications than you could by spending 10 or 20 minutes just providing information and getting into the detail. Thirdly, providing information in different ways and at different levels and layers, which can cater to people with a variety of backgrounds and perspectives depending on parents' needs. We know from other research studies, genomics and otherwise, that there's perhaps a minimum set of information we can provide to parents that they can make a decision without necessarily a huge and endless amount of time, but also ensuring that there's opportunities and easy access routes to find out more, whether that's through accessing more written or online information, decision aids, or being able to speak to someone about any concerns. And finally, knowing that trust takes time and not one point in time. We need to look at consent not as a fixed point where someone's putting something down on a piece of paper, but a process through which parents are hearing about, learning, and having opportunities to discuss information. This could mean an initial approach by a community midwife, then discussion by a research midwife for the pilot, further discussions with family members and others at different points along the pregnancy and newborn journey. This also means thinking about the longer-term implications of the decision made by parents if we're going to be keeping genomic information for a lifetime with that ongoing opportunity to withdraw. For children in the 100,000 Genomes Project or having their genome sequenced as part of the NHS Genomic Medicine Service, there is a commitment to needing young people to reconsent at age 16 or when they have capacity to confirm whether they wish to have their data continue to be accessed for research or not. So through the work already happening in those areas and for this program, we'll need to explore how to best support young people in making that decision. There will be challenges, some of which we'll hopefully discuss this evening and some of which are the topics of future sessions. It can be really difficult to think about the longer term lifelong implications of making a decision like this. We have to think about the impact on the workforce who need time, training, and support to incorporate any new test or process into their day-to-day practice. And there's operational challenges to address in making sure that we can develop digital and other tools to support parents and healthcare professionals to access information, document their decision, and be able to review that at future points if they wish. We'll have opportunities to explore and evaluate this as part of the program and understand what doesn't work and what might work best. So finally, why is this topic important? I assume we're all here because we think this is an important decision to make. It's about families, health, data, and security, and making sure that the parents making these decisions and those that are supporting them can be enabled to do so.
Our next speaker this evening is Dr. Simon Ramsden. Simon is a consultant clinical scientist and lead scientist for rare disease at the Northwest Genomic Laboratory Hub, which is based in Manchester.
So I've got no vested interest in this programme. I've got a very peripheral kind of relationship with Genomic England. I work in an NHS laboratory, and I use WGS in my day-to-day job. And so I thought maybe what I should talk about is really just my perspective about WGS. So we're talking about genetics and genomics, and as we all know, the genome is wrapped up in our chromosomes in the cell, in the nucleus of the cell. And basically what that means is that the chromosomes are basically just a linear, one-dimensional string of information, which includes genes and a lot of other stuff. And what we're looking at here, we're all familiar with. The gene transcribes to RNA, which translates to protein, and mutations in the gene result sometimes in aberrant proteins, and that's what results in genetic disease, the way the proteins work through the body. And that's really fundamentally what we're talking about. Genetic disease is a result of mutations in genes. One haploid genome basically covers all the genes. It's just that we've got it twice, one from mum, one from dad. So we tend to talk in haploid genomes because there's basically one representative genome, that is. If we take that code, that sequence, and just string it out, because it is just a string in these 4 letters, what we find is, what we found when we sequenced the genome was that it was 3,000 million letters long, one haploid genome. So there's 2 of those in every cell. And the surprising thing when we sequenced the genome, we just assumed, because humans are so clever and complicated, we'd have a lot more genes than everything else. And we'd already sequenced flatworms and fruit flies and things like that. And we just assumed that we would have an awful lot more genes than everything else had, because we were so much more clever and complex. Turns out we don't. We've got pretty much the same number of genes as a lot of these other organisms that are much more simple. We actually have about 20,000 genes. Those are protein coding genes through the genome. But beyond that, we don't really understand what all of those genes do. We're still learning every day new descriptions of genes. We now know all the genes, but we're starting to understand them a bit better. In actual fact, only about 5,000 of those genes are linked to human genetic disease. So you could say that actually sitting here talking about genetic disease, we're really only interested in, at most, that 5,000 genes. But if you look at the size of the genes, the 20,000 genes, it's in the white box there. That's what we're looking at. And actually, that's at most what we're looking at. We're actually only looking at, maybe interested in, at most, a quarter of those genes, because they're the genes that are associated with human disease. So we've got this kind of weird thing that we're talking about this evening, which is, why on earth would you sequence all of that information just to get at that little bit of information in the white box, and only a portion of that information, actually? All of that information, it seems like overkill, going through all of that information, sequencing all of that, just to find 5,000 genes. Well, the simple answer to this question, why are we considering it today, is simply because we can. That might sound a bit trite, but it's basically the truth. We can now, we have the technology, for the first time in human history, we have the technology to sequence all of that stuff in one go. And not only that, but at a price which makes it feasible within the context of healthcare. If you look at the price of sequencing a genome, about 20 years ago, when the genome was first sequenced, it cost about £100 million to sequence one genome. The price has dropped. That line, it kind of looks a bit linear, but in actual fact, if you look at the scale down the left-hand side, that's a logarithmic scale. So in actual fact, the price of sequencing has fallen exponentially. So we find ourselves today at a position where the price of a genome, sequencing all of that information, is less than $1,000. You have to take these exact costs with a bit of a pinch of salt. It depends on the salesman that's trying to sell it to you. But I think most people would agree that the cost of a genome now is well under $1,000, £1,000. How does that compare to what I would call a conventional test, the kind of test we've been using all of this time within a healthcare service? Well, the cost of sequence, say 2 to 5 genes, comes out about there. And you can see that those two lines now coincide. And so this is a conversation that it's worth having now. The cost of sequencing all that stuff is now about comparable to sequencing a smaller number of genes. So it's a conversation that we can now have, and it's worth having. And like I said earlier, basically we now have to think about, well, what are the implications of that? What are the implications of sequencing a whole genome? And in the internet world, we love loads of information. More information is better than less. Is that what we're thinking about? Or is it more complicated than that? And so I thought I would just put in a slide of, and I've called this what worries us. Maybe this is what worries me. Not overtly, but these are the kind of things that I think about in my day-to-day job, sequencing genomes. And when I sat down for this event, I tried to think about something, because I'm thinking about these questions all the time. So I tried to think about something outside whole genome sequencing that maybe was a good parallel that would allow me to maybe think about it slightly differently. And what I thought of was the internet. So a lot of the questions that you and I ask ourselves about the internet actually are perfectly good questions to ask about whole genome sequencing. For example, with the internet, can you trust the information? Well, we could ask ourselves that question about whole genome sequencing. And sometimes one has to say that it is not always as we see it. And when we talk about trust, what I really mean is, what's the predictive value of sequencing DNA, sequencing a whole genome? So most of the information we have at our disposal is sequencing affected individuals. And I think one really important question we need to ask ourselves is, how does that extrapolate into unaffected individuals? And we're asking that all the time now. So what is the predictive value of sequencing a genome in an unaffected individual? Second question you might ask, and Amanda touched on this, is, can you limit what you see? And we think about this with the internet all the time. And at its most simple, you could say, well, I can limit it by just typing in a question. If I type in a question into the internet, I'll get an answer back. Or we could think about it at a different level and actually put on some blocks, some barriers to looking at information. Parental controls with the internet is a good example. We can do, and we do exactly the same with whole genome sequencing. We call it virtual gene panels. And I think this is a really important point. We've sequenced 3,000 million letters of DNA. Do you look at it all? Well, nobody will look at it all, because we don't understand what the vast majority of it means anyway. So at its most basic, nobody would look at all of that information. How much of that information do you want to look at? We manage that by virtual gene panels, by actually deciding which bits of the genome to look at and which not to look at. So we've got those same controls that we would use on the internet. Are we getting a balanced view? Well, I know myself, when I'm on the internet, I'll look at certain newspaper sites and I'll avoid other ones. I'll tend to look at the things I'm interested in and probably most likely to agree with, and probably look at less at those that I don't agree with. So certainly my view on the internet is not balanced. I live in an echo chamber, and I think we all do to some degree. Is that a reasonable question to ask about whole genome sequencing? I think it is, because, for example, most genome information we've got And that means all the variability in the genome and all the protein, DNA, phenotype kind of correlations. It's all done in certain ethnic groups. And there are other ethnic groups that are pretty much excluded from our information. We're trying to build those up again, but there's no doubt about it that our knowledge of the internet is biased towards some ethnic groups and against others. That wasn't a conscious decision, it's just the way it's worked out. So it is biased, our knowledge of the genome is biased to a degree, but also whole genome sequencing is itself biased because there are gaps. When you sequence a genome, there are bits you can't sequence using current technology. That will change over time, and maybe we'll touch on that later on. But there are some really important genes that are not detected using current whole genome sequencing. It's important that we understand that if we're going to use this technology. And there are whole kind of categories of mutation that are not detected as well, largely because we don't understand them. We don't understand the consequences of them. So what's the point in looking for them if you don't understand them? You can't interpret them. But it's important to remember that the genome we're looking at is a snapshot of what we understand today. And that's not balanced by any means. So are we sequencing everything? Are we missing things? Might you find something you don't see? And again, Amanda's touched upon this. We worry about that with the internet all the time with our kids, but it's the same question. Are we liable to, when we start delving into the genome, is it possible that we might find things that we don't want to see? We call these secondary findings. And a good example would be if you start looking in cancer genes. In a baby, for example, if you were to look in cancer genes, could you identify stuff that is predictive of disease later in life? It's not always that. It's sometimes more subtle than that. Sometimes even in our gene panels that we've very, very carefully selected for a particular disorder, sometimes in those gene panels, you'll find variants that just you weren't quite expecting, but are predictive of something else. So it's impossible to blank out secondary findings absolutely. It's going to need some careful thought. And with the internet, of course, last but by no means least, privacy and confidentiality is on the top of our minds. And it is with whole genome sequencing. And we're going to talk about that today, I'm sure. A lot very similar kind of concerns around data storage, data sharing, and to what degree can consent accommodate that and get around it. And just as a final slide, I thought I'd take this comparison of the internet to genome sequencing maybe one step further. Just to illustrate, I think one more point, maybe it's the same point. If you ask the internet a very specific question, you expect to get a very specific answer. At this point, I tried to think of a very specific question, and for reasons that I can't quite explain, the question I thought of was, what's the telephone number of Manchester Airport? I thought that was a very specific question, and I thought, well, what will the internet give me? It gives me a very specific answer. But of course, if you change your question so that it's not quite as specific, don't be surprised at what's going to come back to you. And whole genome sequencing is a bit like that. You can ask it very specific questions and stay very safe and get very specific answers back. But if you start broadening out, then it gets far more complicated.
So next up this evening, we have Waheeda Abbas, who is midwifery lead at Northwest Genomic Medicine Service Alliance, and she's a hemoglobinopathy specialist midwife, and she is a counsellor at Manchester University NHS Foundation Trust. And she's also the specialist cultural liaison midwife at the Royal Bolton Hospital.
The question was whole genome sequencing at birth, consenting on and sequencing babies as we know. So basically what this programme is a pilot of newborn whole genome sequencing. And currently we know with the newborn screening programme, there's 9 conditions, that metabolic disorders, sickle cell, thalassemia, hemoglobinopathy disorders. But this pilot is based to expand and allow to be screened for 200 or more rare conditions. And obviously, hand in hand is the genetic data that can also predict persons' risk of adulthood diseases such as heart disease, Parkinson's disease, Alzheimer's, and other conditions that we're aware of. The participation in this programme, it's not fully decided yet, but it could be via blood spot screening programme, cord blood, or which other means they could take the sample. Obviously, it will require parental consent, but there's a lot of concerns around informed choice and parental consent and how will this take place, which is the best way, and the timing of how to obtain this consent and giving the information to the families. Okay, so one of the key factors, it's a critical factor, is communication. And we know how important communication with when you're dealing with the community, the public. And already I feel as midwives that we give too much information to them. It's information overload. And sometimes with the genomics, it's difficult for the health professionals, even though they're doing the job day in, day out, such as screening midwives, they might not necessarily feel as though that's part of genetics or genomics, but it is. So obviously parents would need more information. There would be training involved of the health professionals. Obviously we need to make the parents alert, that they have a choice, it's an option, they don't have to go for it. They need to be informed of what the process is, and what's going to happen with the samples. If they're given that awareness that the samples could be stored, they could be used for research in the future, there's a possibility that you could enhance that engagement. Also, there's quite a lot of studies being taken on newborn spot screening, and the best approach that works is having personalised approach. And we're seeing that with the better births, that we're all talking about personalised care. And I think that's really, really important, because with personalised care, you can kind of adapt to the information at the learning need and the level of understanding, and that can be a great value and an asset to the parents when you're informing about screening or genomics testing. There's a lot of research because, as midwives, we tend to give out a lot of leaflets, which we know once you've got the leaflet, it's probably going in the bin, won't even look at them. So we need to devise other ways. So the digital technology, which is really important, we need to get them platform, because in the future, all the personalised healthcare would be via digital anyway. So why not get the ball rolling and start digital technology so they've got the information, different languages. You can use a QR code, which if once set up, it will take you to that key information that you need to know. And if they've got links to different languages, that's fine, because they can obtain that information. But then you've got other issues like people who use Braille, people who've got learning disabilities. So we need to be looking at services that are more equitable to across board, and that's quite important. And also, because it is genomics, I feel with genomics, it's actually essential that midwives are trained to offer counselling. So with all the questions that are going to be kind of thrown at midwives, they need to know how to answer, or have a lead person who can at least offer these solutions, or are best to advise. The other issues we would say with communication is when obtaining consent. Obviously, sometimes in the newborn screening, normally it's automatically assumed that, you've gone to do the test, and you said, oh, I've come to do the test for your baby, heel prick test. And sometimes the parents don't question and assume that they have to do it because it's normal. But actually, ideally, parents should know what's being offered and what's being screened. Because sometimes when you ask a family, oh, has your baby had the blood test, do you know what they tested for? And they'll say, well, all I know is for some abnormal disorders. And I think that's a gap in the information. I think it's really important that we use interpreters or whatever links is available in different languages so we can get this information across. So I'm just going to discuss about information giving and informed consent. So when we are talking about this part and we need to obtain informed consent, I think Amanda's already touched on this. So when taking the consent, we need to know, that midwives who are taking the sample understand what is the whole process. They understand the pathway, so they can ensure that the parents are very understanding what the process is, and it's easy for them. They need to inform them that the sample being taken, where will it be taken? Will it be taken at home? Will it be taken at hospital? How will it be taken? Because depending on the mode, it's not decided yet. So how the sample, will be analysed in, genomics laboratory, and sometimes, these tests can be used for quality assurance, and also they can be used for research. So these questions are quite crucial. Also, we need to inform that sometimes if the test comes back positive, the GP would be informed. It will go in the child health records and child health information systems. And if the baby is identified as having a positive result for rare disorder, there will be a referral process to specialist services. Also, that I think I mentioned this before, that the blood spot card, the parents need to be aware that they can be stored for a minimum period by the laboratory, and it could be different. I mean, for blood spot card for a metabolic disorder, it could be different time scale to what it would be for the genomics sequencing. Also, it's important that they're informed that, babies' data, if it's used, it'll be anonymized, and it could be used for research studies in the future to improve the care of the families that's offered in the UK. Also, we need to inform the implication of future pregnancies. What would happen if baby was diagnosed with a rare disorder? What would be the options for future pregnancies? Also, they need to be aware that they can decline a test. They don't have to have it, because sometimes the assumption is that they have to have it, but they don't have to have it. They would have their own reason, and this needs to be documented. Documentation is a key. I mean, in some places, they do have consents for vitamin care. It's like a formal process. I mean, use a similar process which says that you have, you've given the information, you've given information leaflet, they've got the contact numbers if they've got further questions. These all need to be beared in mind. And then I was thinking of, so when would the best time that we could ask these families or inform them about genomics testing? So initially, the women the guideline is that the women have the first booking appointment by 10 weeks. So if you can see the first arrow, so 10 weeks, normally what you can do at that point, I mean, you don't have to mention it, but if you can give them a link, for them, or even a leaflet if there's one available, which will point them to, and give them that information on what the test is about, because they don't have to make a decision then. They can just talk with their family, talk with the extended family, talk with their partners, talk to the chaplain or the imam, because they'll have a lot of questions, talk to the midwife if they want more information. And then you kind of leave it until about 20 weeks, because at 20 weeks they have a scan and they'll have the results about, your screening blood results, if they've not already had them. At that point, you could then the midwife could mention it again and just ask the woman, has she had a chance to look at the link, and reinforce that message, give them another link if they've lost the paperwork or they've lost the link, just address it again. And then again, 28 weeks when they come for the research test and the group screening bloods again for the research test and group and save, you could then again mention it again and offer this same information. And by this time they probably have more questions about the test. And then once they've had the baby, when you talk about the postnatal care, pelvic floor exercise, and we're talking about screening, the newborn screening, you can also mention, you know, having a whole genomics sequencing. So there's plenty of opportunities during the pregnancy pathway which could adapt and offer this information. And then if you've documented it, that this information is given, at least then you know that, you know, you provided that information, and then it's just whether they wish to go ahead with the test. And just a final note that, you know, obviously with these tests, sometimes you can have true or false or positive screens. Sometimes parents can be really anxious, and if they don't get a confirmed result within the time scale that you've informed, then it can increase distress in parents, especially if there's lack of education or lack of awareness about the testing. And also challenges around consent, such as what we mentioned, language barriers, religious influences. Also, families can have false expectations, looking at parents' perceptions, and also looking at the vulnerable groups such as, you know, learning disabilities, mental health issues. There's whole challenges out there that, you know, I've not really touched on. So we really need to make effort to increase understanding and awareness among the parents, you know, with and also with the midwives as well. And hopefully, you know, we'll get a good uptake if it goes ahead.
Anneke is Professor of Genomic Medicine at the Nuffield Department of Medicine at the University of Oxford.
So the first question that I have is are we talking really about a research venture or a clinical venture, or probably more likely both, and what implications does that have for consent? As already mentioned, what do we do about the creation of a lifetime resource that this inevitably brings to some extent, and how do we how can consent deal with the longevity of that? And then if we are agreed, which we might not be, but I hope this can come out in the discussion, that consent can't do everything we want it to do in this space, then what are the other things we need to think about? And I think we've already heard about some of those, but they're the 3 points that I wanted to pick up on. But these are some of the recent headlines about this programme. And I think they really bring up that question of whether we're seeking consent for a clinical diagnosis in a baby, i.e. are we looking for an expansion of the current newborn screening programme, or are we trying to do research into the value of analysing the entire genetic code of newborns to provide new insights into disease, or are we trying to do both? And when might we need to distinguish between those two activities? And when might we need to realise that in genomics they are much more entwined or made hybrid than perhaps in some other areas of medicine? And then if we acknowledge that, then when is it realistic to give consent to one or both activities or them joined together? And the idea of genome sequencing at birth is being discussed in these newspaper headlines, certainly as a means to increase the number of diagnoses at birth. So the main discussions that I hear are this is a means to improve the number of diagnoses, so to get up from the current 9 to hundreds. And I think there are two things that are implicit in that message that we could think about. One is that that increase can't be reached by other means, that it's whole genome sequencing that gives us that ability to improve the number of diseases diagnosed. And as Simon has very clearly highlighted, that's not the case at all. We could use other methods to do that. And the other thing that's implicit in there is that early diagnosis or diagnosis at birth will be a good thing. It will give better treatment, better support, better care. And both of those, I think, are important to look at and to check when they are the case and when they aren't. So for example, what do we do about whole genome sequencing, diagnosing genetic conditions that have no treatments, or perhaps worse, aren't a certain diagnosis, or a possibility, or a predisposition, or even a false positive or a false negative? So the chance, I think, of all of these goes up when we're looking for more conditions. And we know very little as yet about the impact of finding false positives or disease susceptibilities in this setting. So if the picture is more about diagnosis and greater certainty and uncertainty, evolving evidence, false positives and negatives are not mentioned so much, then I think we're buying into a discourse about genetics where we imagine it to be like this, that we just simply need to hold up a magnifying glass to the code and a disease diagnosis will leap out of it. And whilst that may be a nice way of simplifying genetics to many people so that its complexities aren't befuddling, at the same time, it means that the possibilities of uncertainty and false positives, etc., aren't as obvious to people. And so one of the reasons, again, it's already been mentioned, but that there's much greater uncertainty in looking at a newborn code is that the context is lost. And perhaps it's a slightly different context to the one that Amanda was talking about, but also a really important context. So if we look at the 100,000 Genomes Project and other studies like so for example, the DDD study have very clearly shown that if you start with a context, with a medical problem or an illness, with signs or symptoms, then using the genetic code to confirm a diagnosis is sometimes really helpful. And the talk of ending a diagnostic odyssey has been much used in that setting. But you can't simply invert that and say, here's a healthy looking newborn, what will this baby get in the future by looking at its genetic code? Because that context of what to look for and how to interpret it is lost. And again, Simon mentioned some of this, but each baby, don't forget, will have about 4 to 5 million variations in their genome and several thousand variations that are rare and that might look, in inverted commas, suspicious in other circumstances. But without the context of an illness, they will be much, much harder to interpret. There's no diagnostic odyssey to end in a newborn baby. So let's be very careful about not conflating the language of the 100,000 Genome Project success and the aims of a newborn project. And then just to talk a bit about creating a resource. So if we do whole genome sequencing at birth, we inevitably, even if we get rid of the data or we don't store it, we will by looking at it once create an audit trail that could be a data resource that might allow all sorts of predictions throughout a lifetime, both by parents during childhood, by the child themselves when they grow older, if it enters their health records, then by others, by their health professionals, but possibly also by insurers or employers, etc., etc. And these are all really important questions about how we could possibly consent and who could possibly consent to the creation of such a resource and how we might access it over time. And once we're talking about the years beyond a newborn diagnosis, I think we're asking consent to do way too much work. And Waheeda highlighted this very nicely. I think we're used to consent in a healthcare setting being for fairly discrete interventions or investigations, knowing the risks and benefit of an operation. Do I want my appendix removed, for example, or my child has a particular problem? I give you, my health professional, consent to do the investigations to diagnose that child. But here in whole genome sequencing research related to clinical practice, we need consent to be much more open-ended. All sorts of potential risks and benefits of others, as others have already highlighted. There are thousands of different rare diseases, susceptibilities, disease risks. It's not possible to go into all of those in detail. And really consent can be about broad principles only. And I don't remember much about the consent I gave to the heel prick test for my 2 daughters, but I think I would have been comfortable with signing up to some broad principles. I didn't need to know about the conditions that the test was looking for, but I consented to diagnoses of treatable conditions at birth. I trusted the NHS to have looked into those conditions sufficiently to decide that there was some merit in me as parent knowing about these diagnoses. But how can we make consent valid to a much, much wider or broader set of possible diagnoses, including untreatable conditions and susceptibilities? And how can we consent to a creation of a resource about an infant who will grow into an independent being, likely to want to have a say over what access and analyses and interpretations will be made? And I think that's a bit more complicated than just looking for reconsent at 16 after consent in the newborn period. So whilst I think there's absolutely nothing wrong with gathering data and thinking about a use for it later, sorry, that's my previous slide, storing a genome as a resource and seeing how that will be useful, I think is a good aim. But I think this aspect needs to be much more transparent in the discussions about this pilot. I imagine that if we discuss the pilot as a research venture rather than as a means to increase the number of newborn diagnoses, the take-up of the pilot might be much less. And that would be a shame in terms of not meeting the pilot's aims, and indeed also in not meeting the aim of improving knowledge of genetic influences in health and disease. But being transparent about what is an offer and behaving in a trustworthy manner, not just relying on people to trust us, but for us to behave in a trustworthy manner, where there can be no accusations of mis-selling something, is also really important where consent can't do all the ethical work in a particular situation. So I'd really like to hear a conversation about what we can do to support consent rather than try to increase the number of information leaflets or tick boxes on a consent form that make us as health professionals feel we've done consent, consent is ticked. Let's talk about the other ethical attributes that are necessary to make this venture a success.
First of all, I'm actually going to take a question that was raised at the previous event, because I said I would, and people hold me to account on these things, so I have to be very careful. So this is a question that came from Michaela Jacobs Pearson, and she asked, how will consent be given for whole genome sequencing, and how will this be transitioned from parent to child, considering the lifelong implications of the data as children reach adulthood? So I think if we go to Amanda for that one first, and then we'll perhaps move to Anneke.
Sure, thank you. That's a really good question, and hopefully I think there's a question that came up in the last session, so hopefully through the talks we've had just now, we've covered some of that. I think it's important that we see, as we've talked about, consent as a process, and that not just around the time of making that initial decision, but also that we ensure that families can stay updated and informed throughout the process of which we're holding on to that data and using it for research. And that means making sure, and I think there were some other questions about this as well, that as those newborns become children and become young adults, that there's a way of involving them in that discussion until the point where they're then able to make a decision themselves if they want to continue to have their data accessed for research. So in the same way that we want to make sure that children are included in decisions about their health, even if the parent's the ultimate person making that decision for them, we need to make sure that we have ways of still keeping them engaged as part of that process. I'll maybe stop there and see if Anneke wants to say anything further. But yeah, hopefully we've also captured some other elements to that through our initial chats.
Yeah, I think what I'd like to add to that is that I think consent looks different in this space, doesn't it? We're used to thinking of it as, well, as long as we've talked about all the risks and benefits, we're used to thinking about it in terms of Montgomery principles, or about just making sure that people are informed or fully informed. We can't do that here. We can't do it once. We can't even do it if we went to try and repeat consent at regular intervals. We have to think of consent as signing up to a broad set of principles, and then we have to define what those principles are and whether those principles include research in the whole consent, or whether we somehow separate those off. And I think it's so important to have these discussions, because on the one hand we've got consent in usual clinical care being sort of one beast, and then we've got whole genome sequencing being perhaps pitched by the media sometimes as more simplistic than it is, or not incorporating all those other factors. So it's really important that we have these sorts of conversations to think about what else do we need when we realize that consent can only give, sort of ensure that people are informed to a certain extent and about broad principles. What do we need to really concentrate on to make sure that the whole venture remains ethical? So whilst we're not so used to doing that in healthcare, I think it's the sort of discussions about transparency and trustworthiness are really important for this venture to be a success.
So I think, Waheeda, people in maternity services would do their bit at the beginning of this process, but I'm not envisaging that they would bow out, I presume, once they'd stopped having contact, because they don't continue to have that contact with the parents. So I suppose, where would that then responsibility move to?
The standard of care was once the baby was born, up to 28 days, they had, the care responsibility of looking after the family. But now they discharged them early, 10, 14 days. And the responsibility then, the care is handed over to the health visitor. But the health visitor is already in the process, because they do a pre-birth visit. So they're already in the process. So I think it's collaborating with your health visitor and working together as a team, making them aware.
And Simon, I sort of wanted to bring you on this one as well, about, because we're sort of are we getting a bit ahead of ourselves, thinking about, as these children reach adulthood, do you, in the lab, with the changes that you've seen over the last decade, envisage that this will be something that, when this child reaches 16, that will be something that you will still be interested in interrogating, in fact?
I think this is, from a lab perspective and a pragmatic perspective, really interesting. I'm interested in making diagnoses and generating the most useful information at any one point in time. And I have to say, from where I'm sitting, if this baby is sequenced at birth and then somebody comes to me, the child arrives at their 18th birthday and wants some more information, I'd be very surprised if I went back to that 18-year-old data. I'd be wanting to generate new data using new methodologies, overcoming some of the practical problems that I mentioned with current technologies. So I think it's a really interesting question, how this data will be used in perpetuity. I'd be surprised if it was used for making diagnostic grade decisions 18 years later. I think there'll be much better technology available in 18 years. What I've seen in the last 18 years is just remarkable. And every year it's an order of magnitude better than the year before. And we're already testing technologies that are infinitely better than the technologies that we're currently using. So in 18 years' time, I think things will be very different. So yes, it will be a data set, and yes, it will be useful. I don't want to predict what it will be useful for.
Okay. Anneke, did you just want to come back on a point there?
Yeah, I mean, I think Simon's absolutely right to highlight that. I suppose what I think needs to come into the conversation is how that data might be misused rather than used to make diagnostic grade predictions. Because if it's there somewhere, it's a bit like doing your internet search and coming out with something that is just not a hit, that you, Simon, might be able to say, well, ignore that finding because that's, you know, that's a website that's now dead, but lots of people wouldn't be able to do that.
Yeah, that's
yeah.
Okay, so we'll move on to some of the questions from this evening. So the most popular one is from an anonymous attendee who, and it sort of leads on from this actually, of what happens when a newborn grows up and finds their genetic information obtained from whole genome sequencing is being used in research, and they object to this and they want their information withdrawn. Will this be possible, and what process will be in place for such situations? So I think perhaps we go to you, Amanda, for that one first.
Sure. The short answer is yes, absolutely. That is the case with the other sort of types of research that happens with genomic testing now, where there's that link to research. So whether that's through the 100,000 Genomes project or in the Genomic Medicine Service, we have processes in place for participants in the 100,000 Genomes project, whether that's when they reach adulthood or at any other point in that time, to withdraw if they want to. And that's a really important tenet on any biobank or genomic related research. And so we'll similarly be needing to think about how we make this a straightforward process when needed for anyone that's part of this program. I think that also comes back to the point I made earlier about us needing to think about how we continue to engage families across time, across whatever the lifetime of this program is, including as children who are part of this program get older and before they have the capacity to make that decision themselves, needing them to think about what the meaning of that could be for them. And that there's always that possibility to withdraw, where then no further information is added into the research library or no further results are fed back to them. So in short answer, yes.
Do any of the other speakers want to add anything to that? Anneke?
Yeah, I think that's right, and it should be like that. But of course, that doesn't answer the question of what's been accessed before that withdrawal happens. Or what's stored in their health records. So I think it is an important question that again speaks to this sort of longevity of what we're testing for here, which is unusual in healthcare. We tend to do investigations and then sometime later do them again, and we might indeed do that here, but there might be a perception that this, once it's done, tells you something about someone for the rest of their life. And the parents might have done all sorts of delving into this code, or the GP might have done all sorts of delving, who knows, into this code before that child is old enough to withdraw their consent. I don't want to be sort of, that's being devil's advocate to some extent, but it's just a bit, I think the question hints at the complexity, which is right to do so.
I think it's sort of, the obvious answer is, is the one that Amanda gave, but yes, but actually with all these things, when you sort of start unraveling it, is actually, as you said, Anneke, it is complex, and all these things need thinking through, which is why it's good we're holding these events and we're sort of thinking about it before the pilot even starts, and the pilot, I'm sure, will help to tease out more of these as well, I hope. So I think I'm going to go for a question now from Therese Callas, which I think it sort of flows on from this a little bit, which is, she says, Anneke raised the important consideration of the uncertainty inherent in future propensity to disease or false positive, negative, etc. And my concern would be identifying an adult onset disease, which the child, once grown up and an adult, may not want to know about, or may not want to have that information shared. So that, I think we'll go to Anneke with that one first, as it came up to something that you said.
Yes, I mean, I share your concern. I suppose the broad principles that we need to devise for this pilot might include, we're not going to filter the whole genome sequence for adult onset conditions as much as we can, and accepting that it might fall out of some investigations and then we know it. But it is possible to say, okay, let's design the set of principles that actually we're only looking for treatable newborn conditions, and we're not looking for adult onset conditions. But we then do still, as you rightly say, Therese, think about who can access the resource that goes into the research part, and whether there is a chance of that information, if it's not filtered out for the diagnostic bit, coming out the research bit in some sort of linkage with other data sets ways.
Amanda, do you want to come in now?
Yes, it's just to build on what Anneke said, really, and to say that part of the work that we're doing and planning for this program is bringing together a working group to establish those exact principles around, you know, on the back of the whole genome. We're not going to look at everything, but what are we going to feed back, and keeping that quite targeted to conditions that are childhood onset. Of course, there may be some uncertainty in that, but where there's some kind of treatment at play and where that treatment has to happen quite soon in order for that to be of benefit, to try and as much as possible keep it quite targeted on conditions that would be relevant at that time, rather than information that, as the person's question said, a child may or may not want to know when they're older.
Okay, I'm going to move on now to the next question, which is from Melody Redman. I think Waheeda could take this one first, perhaps. So, is there any evidence about any effects of uncertain findings on parent-baby bonding? Do we have any estimates on how frequently variants of uncertain significance might be found? So I think if you take the first part of that question, Waheeda, about, I suppose you might have sort of, you know, not papers as such, but, you know, from your experience about any impact on that relationship right at the beginning at that very special time.
Actually, I've been in a situation where a baby was, when it was delivered, it had some, you could see there was some deformities. So in terms of attachment, I didn't see such attachment issues. In terms of, they just wanted to know what was the best what they could do for their baby in order to kind of improve their care or to help alleviate the deformities. So basically just wanted to see what was on offer and how could their problems be overcome. And in another situation where once I did the blood spot screening, and you know, I'd gone through all that information, and I think if as a midwife you give all that information, which is really, really crucial, because then they're prepared. And in this case, this baby had hypothyroidism. A week later, I think it was about a week later, when I found out that the baby had, you know, a positive result, I actually went to see the family, and they were actually quite thankful that, because they were aware about, they didn't panic because they discussed it and they researched it. But not everybody is like that. Not everybody would research it. So it's more about preparing for these vulnerable families who might not have the necessity, that capacity to think what would they do if the result was positive. Because majority of the time, even myself, you think it's never going to happen to you, and you're kind of in that process thinking it's never going to happen to you. And then when it does, they kind of panic and they go into the realms of denial. And when they're in the realms of denial, they could delay treatment, which could be really important.
Okay, who wants to pick up on this next? Amanda, do you want to come in there?
Yes, please. I think the question and what Waheeda was saying really speaks to the support that we have to make sure is in place, whether that's genetic counselling, support from midwives, health visitors, charities, etc., that's needed when those results come back. And also, as some may be aware, but there's another study called the BabySeq study that was done in the States, and that did genome-wide sequencing in the newborn period as well, feeding back on a large number of conditions as part of a randomized clinical trial. And they recently published a paper that showed there was no kind of persistent negative psychosocial effects on families that got the finding versus those who did not. That being said, it's worth noting that that's one paper and one study, and it was quite a small sample size. But it's similarly a question I think is something that we're going to explore in the pilot in terms of, for those families that get their results, one, make sure we plan up front to ensure that they could get access to support that they might need, and two, be able to explore what that journey is like for them, which, as Waheeda I think was alluding to, can be very different based on families who are maybe more or less vulnerable or have more or less support already available to them.
Simon or Anneke, do you want to come in on the point about, do we have any estimates on how frequently variants of uncertain significance might be found?
How frequently they might be found?
Yes, that was the, yes.
Depends how you apply a filter, doesn't it?
Sorry?
It depends what filters you apply.
Yeah, exactly. It's important. How long is a piece of string? It depends how you analyze the genome. You could really keep it down to virtually zero if you wanted. It's impossible to answer, really.
It is a good question to highlight.
Good.
Yeah, and to highlight the fact that our knowledge of the genome is still evolving. And so what we're uncertain about today might be much clearer in 5 years' time. So a VUS today may be a benign polymorphism next year, or a known diagnosis of a rare disease.
Yeah, and it works both ways. You know, some mutations are pathogenic it's rare, but some mutations are pathogenic in some groups and yet not in others.
Yeah,
and you know, we'll learn more about that over the years as well.
Okay, so we'll move on now. And there's another question from someone who wanted to remain anonymous. In the current heel prick test, whilst I agree current counselling is very limited, all these tests meet recognised screening criteria, particularly early diagnosis leads to improved health outcomes. Is thought being given to creating whole genome screening panels which meet current screening test criteria, which that parents could consent to, but opt out of those with current treatments or benefits of earlier diagnosis? So Amanda, do you want to go first on that? And then we'll perhaps go to Anneke.
Yes, sure. So in part, this I think goes back to this working group that we've brought together with experts from both the clinical science, ethical, patient, public views on how to make those principles of what we should include. We know that the UK National Screening Committee has criteria for selecting conditions they would screen for in a clinical context. Given that what we're talking about here is a research pilot, we still need to think carefully about what those principles are that underlie the conditions we select, and namely those that are of childhood onset and actionable in childhood. So I think ultimately those criteria that the Newborn Screening Committee and we use will be largely similar, but noting that theirs is in a clinical context, whereas ours is in the context of a research project. So we'd have the ability to potentially explore more broadly to some extent, and that would be looking for potential larger number of conditions. That question around whether parents might be able to make a choice to have feedback on some conditions than others, it's one that I think we would need to explore, and also think about whether, if we are giving that choice, what's the basis on which we've put some conditions into one group versus into another group, and making sure, because there's so much gray area in that, if we're going to be splitting up that choice, we'd have to think about why that is and where parents might make different decisions, I suppose. But keen to hear other panelists' thoughts on that.
Yeah, I think for me that highlights that clinical research boundary so beautifully, doesn't it? That people who work in genomics come across every day, that, you know, some activities we might do are really purely clinical and some are purely research, but an awful lot of what we do is a bit of both. So, you know, so how can you research variation in the genome to try and find out better diagnoses for the future and not introduce a whole load of uncertainty? And keep to the principles of only looking for conditions that sort of fit the current newborn screening criteria, where there is some evidence of benefit that you find them early. So I think that is just a really difficult one to get right. And I don't really know what the answer here is, apart from that I think much better discussion about the fact that there is a research element to this, I think would be helpful, because I too often hear it, and I think Genomics England is very good at saying this is research, but the coverage in the media is very much about increasing the number of diagnoses, and that more is better, that sort of discourse. And surely it's got to be better to diagnose 400 conditions than 9 conditions. Well, I suppose I'd say two things to that. Maybe not. Maybe most of those 400 conditions are not really worth diagnosing because they don't meet those criteria. And secondly, I think it's also worth thinking about most of those extra conditions might be incredibly rare. So the absolute increase in numbers diagnosed per year might be relatively small.
Simon, did you want to come in? I wasn't sure.
No. Well, yes. I'm not on mute now. You can hear me. Okay. It goes back to this question about the research diagnostic boundary, doesn't it? It's worth saying that the UK National Screening Committee recommendations are quite conservative, and the numbers of tests included in the UK is really very small, particularly when you look at other countries. So there are some easy, quick gains that would stay well within diagnostics. Looking at other countries, what genes did they? I think in America they used similar, not exactly the same criteria, and test for 200 genes, something like that. So one could expand the screening, and not enter into research, I think, and that's an interesting question. And certainly part of my job, but I'm really interested in how far could you expand the screening to the benefit of the baby and stay within diagnostics and within the remit of a perhaps broader screening committee remit. Beyond that, you get into research. So I think there is room for expansion and staying within diagnostics, and then we're into a different area, which is what we're touching upon.
And I think those views about the current system certainly came out quite strongly at the event that we did on the current situation, that there was a sort of an appetite for it to be expanded. So we're going to move on now to epigenetics. Someone wants to know, what about epigenetics and environmental factors that influence genetic expression? Is diagnosis and research not made less definitive or exact by these other external factors? If whole genome sequencing is done at birth, whether many external influences may influence genes as the child develops. So how certain or useful are the results as they grow up and do various things, or fed whatever and have different life experiences? So perhaps we'll put that one on Anneke first because she's smiling. So I'll go to her.
Teach me not to smile. Yeah. That's a really good question, and I think it speaks to that slide that I put up of, magnifying glass held to the code, where people expect a disease to leap out, and actually, it's, as you rightly say, it's so much more complicated than that, isn't it? There's all these other factors, environmental, epigenetic factors, that mean that if you haven't got the context of signs and symptoms in front of you, that the genetic code that you think predicts the disease is less predictable than if you've got that context. So the context will tell you that there are environmental, epigenetic, and genetic factors working together to give you a disease. And if you then only find one of them in a healthy child, that's just turning it upside down and not being able to do the same thing. But whilst we have very sort of shiny techie views about genetics and we don't recognize that will be a misunderstanding that happens. I think it's really important to keep highlighting that.
Did anybody else want to come in on epigenetics? Simon?
Well, just to say that it depends upon the sequencing technology that's adopted, but I would hasten to guess that whatever is adopted for this project won't incorporate epigenetics. I would also hasten to guess that, going back to my earlier point, in 18 years' time, I suspect we will have that technology, and it will be useful. So it does, again, raise the question, you know, that whatever we generate today is of limited value. I'm not saying it's useless at all. It's got a huge value, but it, as Anneke says, it doesn't incorporate all that other stuff. And I think that's where we're going to find most of the changes over the coming years. We're going to look at proteomics. We're going to look at epigenetics, all of those other factors that influence gene expression and give us a better predictive guess at phenotype. Of course, you won't get that information out of this project. So, yeah, I look for that's the way it's going to go. So I don't doubt it.
Okay. So we have a question that sort of often pops up in relation. I think it popped up quite a lot in relation to the 100,000 Genomes project. And it's a question from Jan Kirshner. It's not really possible to anonymize whole genome sequencing data, as the data is unique for each person. How do you solve this for later research use? So perhaps, Amanda, you want to go first on that one?
Sure, that's correct. You're right, there's always the potential for re-identification. So we would need to consider that genomic data and in that sort of research environment is de-identified rather than fully anonymous. And that's also important so that we have the ability to link back any research findings to the patient's clinical care, which should be part of that benefit from research, but we'd have to make sure that that's done safely. And there's a lot of checks and balances and that need to be put in place, and rightfully so. And that includes things like having access committees on deciding who can look at the data, and that's for health purposes, that people can only look at that data in a sort of aggregate way in a monitored and sort of secure environment, and more like people have described it as kind of like a lending library or an aquarium where you can look at things, but you can't take them home with you. And then also checks before any information is taken out, which again would need to be a sort of aggregate or summary data rather than individual data, unless there's been kind of explicit consent to do that. So hence, yeah, we'd consider that kind of de-identified rather than fully anonymous, but need to put those things in place for that reason.
So unless any of the panel have got anything pressing to add on that, I'm going to move on because we seem to have a lot of questions in the box. So I'll try and crack on with the next one, which is from someone who wanted to be anonymous, which is how will the effect of results of whole genome sequencing on genetic relatives be dealt with? Will consent to know results and/or genetic counselling be provided to genetic relatives as well? You know, you could see lots of scenarios when this may occur. For example, I suppose if you've already got existing children who haven't benefited from this, and you find something and you might want to have them tested, how would that work in practice, and what thoughts have been given to it? And what do we do at the moment? Because these things obviously pop up. So who wants to tackle this one first? Perhaps we'll go to Anneke in the first instance.
So smiling again, Sarah. Yes, I think that's really interesting. And of course, in genetics, we're used to thinking about relatives. We deal with families rather than individuals. And it's really important that when you make a genetic diagnosis in one person that might have implications for relatives, that we think about those relatives. I think that to some extent we can make an argument that a genetic diagnosis in a family is actually a familial one rather than an individual one, and you may have found it in a person first, but that doesn't mean they have a right of veto over using that result in others. And I think that in genetics, we're good at making that explicit at the outset and saying that your result might be useful for family members. But I think there's a really important question there about what happens when genetics and genomics becomes much more mainstream and routine. Will that consideration have been made by people? I think that a lot of people are very savvy about this. So, for example, a lot of people that I see in clinic would be much more comfortable with the sharing of their genetic information with relatives through some means, not necessarily directly, or they may not be the ones to want to do that. So they may be much more comfortable with that than a lot of professionals are who worry about what consent is needed and who owns the data, etc., etc. But I think there's a lot more to be done there. As a slight sort of tangent in answering your question, it is interesting that the pilot for whole genome sequencing has gone just for newborns rather than for trio testing, which of course would be much more informative. So by trio testing, I mean to compare the sequence of the baby with both parents, because then you can rule out a lot of uncertain variation. If it's present in a healthy parent, you can say, well, actually, this is not going to be anything. And I presume that's for all sorts of practical reasons, but maybe also those reasons that the complexities of then finding things in parents when you're setting out to diagnose newborn conditions is perhaps just too great for a pilot.
Yeah, I think Anneke's right there. It's while we haven't yet, I think, confirmed which samples we're going to take and how that's going to work, it is a careful balance between the benefits that getting a child's parent samples up front can give you in terms of not having so many genetic changes that you can't interpret very well and being able to understand that a bit better, and taking all of that up front, having to store and do something with all of those samples when a relatively small proportion will have a meaningful result. So it's a sort of tricky balance that we'll have to manage for the pilot and see how that pans out in terms of what would be most useful in the future, I guess.
So next question is from David Curtis. How can informed consent be valid if parents are not given quantitative estimates of likely outputs, i.e. probability of discovering actionable disease is X rather than we might detect an actionable disease? So I don't know who wants to go on that one. I'm going to go with Amanda first.
Sure. Thank you, David, for your question. I think that goes back to what I and Anneke and I think others have said about the concept of broad consent and how we have to think about this in the context of genomics. If we can't accurately provide a specific percentage risk about what will happen, I think we shouldn't, because I think it'd be more risky to try and give that estimate when we can't be accurate on what that could be. I think in order to help a decision be more informed, we have to bring it back to what the principles are by which a family is able to make that decision and think about how they might respond and react to the potential findings that we could have. We can be honest about the fact that for most families, that chance of finding anything will be really, really small. And that for some families, maybe with a family history, we can tailor our discussion a bit more if we think that there's a reason that we should be more highly suspicious of a condition coming up in them. So I think in general, we need to, in order for that consent discussion to be more informed, we have to be more broad in our approach of getting families to understand the sort of general, more uncertain nature of what we see when we talk about genomics.
Does anybody else want to come in on that, Anneke?
Yeah, I think it's such an interesting question, but of course, I think, Amanda, you said this in your talk, but consent is not consent if it's not informed. So why we always want to add informed in front of the word consent is an interesting one. It's and I take it to mean that we're trying to be very specific. We're saying here, we want the consent to be particularly informed about these particular risks. So I think a better adjective might be specific consent. And that's one that we're all very used to in healthcare. We take a lot of specific consent from people, and we might give specific consent using the internet analogy to, to allow cookies on a particular website or whatever. But we can't do that here. We just can't use specific consent for this ongoing lifetime resource. So that's why I started by suggesting that consent can't do all the work here. And we must learn to stop trying to make it do all the work, because it just won't happen. And one solution might be to say, okay, well, if we can't do all the work, then we shouldn't be doing it at all. But I think there might be another solution that says, as Amanda and others have said, consent here is about signing up to a broad set of principles, and we need to work really hard on what those broad principles are, but it's no more specific than that. But that takes place in an environment that is transparent, trustworthy, and can be trusted, because trustworthy and trusted aren't necessarily directly related. So I just think it's time now that we stop thinking of ethics as being just about consent, which there is a tendency to do, and think about the other ethical attributes that are needed in this space.
I think that ties into something that actually is another question from David Curtis, but I'm not going to put it as a question, but he sort of makes a comment really that the proposed trial is a research project, not healthcare. So ethical standards for it need to conform to those for medical research. And which is then used the phrase you'd like us to move away from, which means fully informed consent, not just trust the doctor. So I wasn't sure whether Simon wanted to come in or not.
Oh, sorry, I've got that look on my face. I think.
Sorry.
Can you hear me? Yeah. Yeah. Okay. I think what I was thinking, and I've not really thought it through, but it goes back to what I was saying, is that this is just a test. There's nothing particularly clever about WGS. It's just a test. It's what you do with it that's the trick. And as a test, there is a lot you can do in this space that has huge utility in terms of neonatal screening and the kind of the diseases you can screen for, avoiding or curing diseases. And what we have to be careful is that we don't disadvantage people who don't want to take that next step and enter this realm, which may be an uncertain one or maybe less uncertain, depending on how it's being counselled or how you perceive it. But I would hate to think that they were disadvantaged because they didn't want to take that extra step, because the technology has huge potential to improve the outcome of these babies.
Thanks, Simon. So the next question is from Nicole MPH. So how would you handle reanalysis and is it recuration? Will providers be able to order reanalysis with new phenotypes? And what about the recuration of variants over time?
We're currently sequencing genomes in the context of affected individuals, and we have exactly the same question. How often do we go back when we find something that's not quite interpretable or we don't quite understand it? How often we go back to it? And by recuration, I suspect what we mean is how often do we go back to consider whether or not we can reclassify it within the context of this disease? Is it causative? Is it pathogenic? That kind of thing. So we're thinking about these all the time, and within the context of a presenting phenotype, I guess you might go back every time the phenotype changes, or maybe every time the patient comes back to clinic. So we have kind of measures currently where we can think about that. Of course, this is a research program, and I don't know what element, how this will be written into the proposal, so I can't answer it in the context of this project.
Does anyone else want to come in on that one before we move on? Amanda.
I'll maybe just briefly note that it's definitely that question about reanalysis and sort of re-looking at what we know about those gene changes is a really important one. And I think it's part of what might be needed to make having a genome sequence useful over the course of a lifetime is we need to have the ability to dip back into that to be able to answer other types of questions. So it's something that we're kind of looking into in terms of the practicalities of that. And then there's obviously the ethical questions on that about when and whether and what we should reanalyze, and also can we.
And we're not that good at doing that in our healthcare at the moment. So people who come to genetic clinics for a diagnosis or an estimation of their risk, a lot has been written in the literature about, is there a duty to recontact and to reanalyze? And I think so far the answer has been, well, there might be if we had enough resources, but the resources don't go anywhere near dealing with what we've got in front of us. So it's all a bit haphazard. But it's been that sort of discussion. And I think it's even harder in a program of newborns where you're not starting with that clinical question of what is wrong with me or what is my risk. You're saying, we're doing a public health screening programme, and how often and when should we go back and look at all that data again that wasn't in the initial filter? I think it's a really good question. And I think it all comes down to that same thing again of this is a different type of investigation. It's not an X-ray that we're repeating or a blood test that we're repeating. It's something that tells you about someone's genetic makeup, that is a lens into their future, but not a terribly accurate one.
Okay, so we're getting quite close to the end now, so I might just try and bundle a couple of questions together. So there's a question about the level of engagement with maternity and midwives on this work, and how it's going to be able to work for them in practice. So I think we'll go to Waheeda first to start to answer that, but also I think as part of that, there's a question from Marlene Jansen how will you assess the information need of the parents for the informed decision? Because I think Waheeda sort of encompassed both on that. So if you start first and then we go to Amanda.
Okay, thank you. So basically, in terms of the understanding of the parents, it's individual assessment, because in community midwives, ideally you should have your own caseload. So you are developing that relationship with that woman from the day they book with you up until they deliver, and then postnatally. So ideally you should have that relationship with that woman and be able to give them that confidence and build that trust. And if you can't answer any questions, find a specialist who can provide that information to give that confidence, and so they can give informed consent, because that's quite crucial, because it's not as simple as just doing the test. There's more to it. You've got to think of the implications about it, the outcomes, and then what the knock-on effects can be from that if it's an undesired outcome. So that's what was it, the earlier question? And
then the other part was about how the level of engagement with people like yourself on the work, and how this will work in practice.
Well, ideally, I think it's really important that the midwives are made aware, they attend training, cascade training. There's a lot of e-learning. I mean, the issue is with the time because of the COVID, and also looking at staff capacity. So it's about building in the knowledge which is easy accessible, so such as e-learning, which can help, and and they don't have to be on the shop floor. They can just take an hour out and learn about it. Also, not all midwives will need to know about genomics. I mean, they just need the basic knowledge about who to refer to, where to signpost, and how to provide that information. And also about, I think it's really important to, once this pilot is given a go ahead, assuming, there'll be a certain amount of hospitals that will pilot it, because you can only pilot it, say, like 10 units across the Northwest. That's just an example. But actually, working with a maternity group, maternity voices, getting them involved. So getting the women involved, voicing their concerns, and just seeing how they can overcome any hurdles, because the patient's perception is really important. So they need to be involved in the whole process, have them on the board, invite them to the meetings, so you can look at their perspective.
And Amanda, is that something that Genomics England are planning?
Yeah, definitely. We've actually just started a working group on the recruitment pathway, which includes people like Waheeda mentioned, midwives, maternity voices, from different sort of patient and public groups. And it's chaired by a member of our participant panel who herself is a patient with a rare condition. It's important that we involve those individuals with different perspectives while we're making those decisions, rather than sort of checking with them later. So that's the kind of model that we really want to make sure that we keep doing, because it's really important that all of those people feel they have a real stake in this. So like Waheeda was saying, with midwives and people in maternity care, if a lot of this is going to fall to them to have to learn and support patients and parents through this, then they have to be part of that decision making for sure.